Welcome again to this presentation. We're coming to almost 80% of the module by this coverage. And so I would ask that we are able to try as best as possible to catch up. The part we are going to deal with is part C, which will then transit into part D. And we will definitely be dealing with the breeding strategies of important crops. And the specified objectives are here from 1 to 7. Look at them closely as you listen to this presentation. This presentation will be sent as a PDF form. We are at this point of our focus. As you can see on the bottom where the arrows lead to, we have the colored portion, which we are going to be looking at as we go on. I'd like to quickly come to just the introductory part of this, and you will excuse the word restriction is misspelled here. But again, population restriction is as we keep on selecting and selecting, and we retain the progenies. Remember, we undertake a population restriction. And here, this is a fact of life, particularly because it is a consequence. The size reduces or restricts as a consequence of genetic drift. It can also reduce because of the domestication and inbreeding. Now, despite these restrictions, the size is reducing, that is to say, the intention in plant breeding is to, every time we advance a generation, is to capture a variation in successive selections as much as possible from the germplasm or its mutant stage till the final target product we are heading for. So in this regard, plant breeding is the capture exercise of heritable genetic effects or heritable variation as revealed by the VA statistics, as will become evident as we move on. Now, we are particularly interested in traits that we can actually improve in the subsequent selection, which are related to fitness. When we talk about fitness, remember, we are interested in reproductive fitness. And reproductive fitness that is high fitness is adaptive. And so this fitness may actually increase the VA statistic due to contributions that come from dominance and epistatic variances. Dominant and epistatic variances are a little tricky because they are not heritable in real sense of what we are looking for as breeders. So here, when we are restricting the size, that's a selection process by which then certain desired traits we expect when they're heritable, they'll become more prevalent than other traits that we are not paying attention to due to the genes found on the species chromosomes. I want you to remember this, that when we talk about traits, they are determined by genes or alleles. Those are the alleles that code for the traits which show the characteristics that we are able to observe in the phenotypes that we work with to select. Which brings us to a very important aspect that is going to be running right through the rest of our aspect of plant breeding from this point on. That when we talk about variation across, particularly continuous variation, then obviously we are talking about quantitative loci, which have multiple alleles or genes that come in various forms and will experience selection. It is worth noting here that a breeder's key or primary interest is in obtaining a superior selection response. Obtaining a superior selection response from crosses that he or she has made between or among good parents, aimed at combining the good alleles that each parent may be carrying as trait determining. So plant breeders will keep crossing and continuously selecting better than the previous traits, further and further until more superior traits are incorporated into a final plant variety than what either of the parents originally showed. The parents may actually be already existing elite varieties, but as I've mentioned before, plant breeding is a continuous updating, just like the updates we get on computer. You get a certain software, and when we have the software, so we have the windows, the browsers, they must be continuously updated, removing certain unwanted artifacts like the viruses and so on and so forth, and making the existing program or application even much better than the parent one. That's exactly what happens when we are involved in crossing and continuously selecting the subsequent offspring so that we end up with a variety that is new. So really, that variety that is new is in effect a selection response. Let's call it a selection response for now. Because at each successive evaluation, we are evaluating a selection response, not necessarily a new variety. So that response depends on the VA. That VA, particularly with that subscript A, represents additive variance. You can think of the V sub A as several genes adding to a genetic effect, hence additive. So to emphasize again here, remember that additive genetic variance is the only part of the genetic variance that is heritable. That we must emphasize. And it is the one that is contributing to selection response. Now, this VA can actually, for a quantitative trait, can actually increase even as we bottleneck the size of the population. Remember, as we restrict it, start with a broad population, and as we go a little further and further and further, we constrict it. That doesn't change the VA for quantitative trait. So this increase in VA is actually very important because then that component may enhance selection response. Now, this potential gain from inbreeding has very strong implications for crop improvement programs. So in this unit, in this particular unit, we want to give a due attention to selection response in detail. To do this, we have some basic quantitative genetics that we need to look at as making breeders' favorite equation functional, which takes us to the issue of quantitative trait loci. I think by now you should understand this better, coming with the fact that we are interested in quantitative variation, and this quantitative variation may involve multiple alleles that are traits or are trait determining. So there are characteristics of QTLs. One, controlled by very many genetic loci, and two, exhibiting a wide range of variation. But again, we have to realize that variation may be due to genetic and environmental effects. They will always be there. So really, when we do have the genes that are influencing quantitative traits, we now have had them called quantitative trait loci. You understand? Do you understand? We have QTLs in effect, which can be determined, can be determined through QTL analysis. But I want to promise you, it's expensive, it's labor intensive, it is fraught with statistical problems, and you need fairly sophisticated equipment and lab. So it's not very easy at Ogongo campus in our department at the moment to have a QTL lab. But if we had one, we can do what is referred to as QTL mapping. And that QTL mapping can reveal three important things. One, can reveal the number of loci that influence a QT, a particular quantitative trait, a number of loci. Remember, locus is single, loci are plural. Can reveal the magnitude or the order of the effects on the phenotype. Can even reveal the location on genome, which can be done through what is referred to as genotype by sequencing. Now, we did mention something very key here, that we are interested in the V sub A, the additive variation. It must be what we aim for because it is the portion or the variance that can be inherited or is heritable when we are crossing two parents to offer or to transfer or to move what is heritable variation. So it brings us to the concept of heritability. Now, let us remember our normally distributed population that we have seen in the previous lecture, where we saw a bell-cup shape, a bell-shaped curve. On the left, having shorter people or shorter plants, height, and on the right, having taller plants. And we saw that shorter plants or shorter height involves fewer genes, and taller height involves more genes to actually determine the tall height. This is very important. So here, heritability is the proportion of the total phenotypic variation that we may look at in considering the character here, the character value across. And the frequency is simply the counts we see of the individuals that we may actually measure across the normally distributed population. Then from that, we can estimate heritability. So I would like us to quickly listen to this bit here, and then we can understand a little more about.
Hi everyone, welcome back to Useful Genetics. This is lecture 9B. We're going to extend our discussion of heritability in the previous lecture to how can we actually estimate heritability. We'll talk about how to estimate it in breeding studies of plants and animals, a way to estimate it for ecological and evolutionary studies, and how to estimate it in humans when we can't actually control what humans do. So we need to find ways to separate the effects of genes and environment when we don't know which genes matter or which aspects of the environment matter. There are several ways we can do this. One is to measure a population's response to a single generation of defined artificial selection. Another is to remove the environmental variation completely, as much as possible. And the third is to compare the observed resemblance between relatives whose phenotypes we've examined to their predicted genetic relatedness. And I'll discuss all three of these. So first, for breeding studies, it's very important to know what the heritability is before doing the work. So one way to do that is to do one generation of the work and very carefully measure the response to selection. So imagine you have, say, a large breeding colony of pigs, and you want to select for greater weight at 6 months of age. And so you take your population and you carefully measure the distribution of weights at 6 months of age. And then you breed only from the pigs who have high weights at 6 months. These are the desirable pigs. If you find that in the next generation, when you again carefully measure the distribution of weights at 6 months, that their distribution is like that of the whole parent colony, you'd say, oh, heritability is very low, maybe it's 0. We had better work on improving the nutritional status of the pigs instead. If you find that heritability is extremely high, you see that the offspring population has the same distribution as their parents do, then you would conclude that heritability was very high and the breeding program was likely to be very successful. More likely what you find is that there is some heritability for height, that by selecting high weight parents, you produced offspring whose weights were definitely higher than the original full population, not as high as their parents. That tells you that heritability is somewhere in the middle. Now, in ecology studies, you don't have defined populations, but it's possible to do a different kind of experiment. You can take subpopulations from different environments with different phenotypes and find out to what extent the phenotypic differences are due to genetic differences rather than to environmental differences by transferring a population to a controlled common environment. Here we've taken seeds from plants whose phenotypes were tall in one environment, short in another, and we're growing them in a common greenhouse. In plant biology, this is often called a common garden experiment. And it lets you discover how much of the phenotypic difference is due to genetic differences. Now, in humans, we can't do these kinds of experiments. Instead, we have to work with whatever information normal human behavior has given us. And so an excellent strategy that's very widely used is to compare the phenotypic similarities of relatives to the similarity that would be expected if the trait were perfectly heritable. And you can do this because we can calculate how genetically similar we expect known relatives to be. We'll talk more about this in a later lecture in this module. But for now, here's some basic information. For identical twins, the expected relatedness is 100%. They're genetically identical. And the similarity of their environment is quite high. We can contrast identical twins with fraternal twins. They have only 50% of their genes in common, but their environment that they grow up in is very similar to the level of similarity between the environments of fraternal twins is very high, like the level of similarity of the environments of identical twins. That lets us see the effect of different levels of relatedness with the environment as constant as we can keep it. Another way to do this kind of study would be to compare, say, fraternal twins, siblings, and parent and child, all of which are expected to be 50% identical. So fraternal twins are 50% identical, as are siblings, as are a parent and a child. But the levels of similarity in their environments are quite different. Siblings, because they're different ages, will have moderately different experiences within the family, and parents and child typically have quite a lot of difference in their environments. And these kinds of experiments can let researchers fish out the genetic components and the environmental components of phenotypic traits that they're interested in. So we've talked about different ways that heritability can be estimated, even though the genes and environmental factors responsible are completely unknown. In breeding projects, it's wise to do one generation of breeding with selection and carefully evaluate how much effect the selection has. In ecological and evolutionary studies, you can rear populations that have different phenotypes in a common environment, ideally one that you can control. And for human studies, the usual strategy is to carefully compare the level of phenotypic relatedness with the predicted level of genetic relatedness. Coming up next, going to go through a couple of examples of studies showing the heritability of human traits. I hope to see you there.